a good day and thank you for joining. Today I'll be presenting the study titled Integrated Multi-Omics Analysis of Lower-Grade Glioma using data from the Genome Atlas. So this study actually brings together somatic mutation profiling, transcriptome-wide differential expression, and pathway enrichment across a number of tumor samples from the TCGA database. So the central finding is the identification of IFNA6, a type 1 interferon gene at the chromosome 9 as a biologically coherent and clinically actionable candidate biomarker. Let us begin. So basically, the study overview. Before we go into the individual analysis, here is the overall framework of the study. So we walk across these six streams. So the first was the somatic mutation analysis of 510 TCGA lower grade glioma tumor samples using MAF files and MAF tools. Secondly is the batch effect correction, whereby we merge the TCGA tumor expansion data, the RNA expression data with 150 GTEx normal brain samples and applied combat seek to remove inter-cohort technical variation before any downstream analysis. Third is the differential expression analysis using DCQ across all 51,001 gene transcripts. Fourth is the pathway enrichment using GAGE across 177 human CAC pathways. Fifth is the biomarker prioritization by cross-referencing our filtered differential express gene list against comparative toxicogenomic database, glioma gene set. And sixth, the key finding, which is IFNA6, which emerged from the prioritization method with genome-wide statistical significance. So I'll proceed with the introduction. So lower grade gliomas account for approximately 80% of malignant central nervous system tumors in adults. They are a heterogeneous group consisting of WHO grade 2 and 3 diffuse gliomas that share a descriptive clinical course. Grade 2 tumors have a median survival of 5 to 10 years. Grade 3 tumors, 2 to 5 years, but these numbers mask enormous variability depending on molecular subtype. So the clinical challenge is for multimodal treatment, which includes surgery, radiotherapy, and chemotherapy, consistently results in recurrence and malignant progression. And when checkpoint immunotherapy has been tried in unselected glioblastoma patients, it has failed to provide long-term benefit. That last point is critical because it motivates the central question of the study. Can we identify who should receive immunotherapy and why it fails in others? So the molecular taxonomy of the LGGE database. So the 2015 TCGA lower grade glioma landmark paper established that the lower grade gliomas fall into 3 molecular subtypes based on 2 biomarkers, which are IDH mutation stages, IDH mutation status, and 1p19q fold deletion. So track 1, as we see in the middle, is the astrocytoma lineage, IDH mutant 1p19q intact with TP53 and ATRX alterations. Track 2 is the oligodendroglioma lineage on the left, which is IDH mutant with 1p19q co-deletion, CIC, and FUBP1 mutations. And then the IDH wild-type tumors, which are on the right, which behave molecularly like glioblastoma and carry the worst prognosis. These subtypes are now mandatory classification criteria under the 2021 WHO CNS classification. Over 90% of IDH1 mutations in lower grade glioma occur at a single amino acid position, which is at arginine 132. Why does one point mutation matter so much, especially in terms of IDH? So the IDH1 R132H substitution converts a metabolic enzyme into D2-hydroxyglutaric factory. This D2HG competitively inhibits TET2 DNA demethylases and KDM histone demethylases. That inhibition triggers genome-wide CpG hypermethylation, hypermethylation, which is the G-CIMP phenotype, which silences hundreds to thousands of regulatory elements simultaneously. So the downstream consequences is a catastrophic genome-wide collapse of the normal brain transcriptome. And critically, vorasidenib, an IDH inhibitor approved in 2023, targets this exact mechanism, which is why restoring suppressed programs through IDH inhibition is now a therapeutic reality rather than a hypothesis. So the research gap and study aims with regards to this study. So as we can see on the left, the knowledge gaps are four that we have identified. First, the LGG transcriptome has not been systematically characterized at genome-wide scale using a unified analytical framework. Second, no integrated multi-omics approach has identified the subset of dysregulated genes with the strongest disease relevance. Third, the relationship between transcriptomal reprogramming and immune gene dysregulation remains poorly defined. Fourth, interference signaling is almost entirely absent from the lower grade glioma transcriptomic literature. On the right is the main study aim, which is to develop an integrated molecular framework linking IDH-driven epigenetic remodeling, large-scale transcriptional reprogramming, and immune-related dysregulation in lower-grade glioma. So to get this will constitute an integrated multi-omics analysis that uses the full analytical power of the TCGA LGG public resource. And the methodology here, which I will get into next, developed provides a replicable framework for addressing these gaps in future work. So the methodology, so we will start with the data sources. So we use four data streams: somatic mutation data, which are the 510 tumor samples downloaded as MAA files from the GTC portal with only past filtered variants retained. Then we downloaded the RNA-seq expression data from the TCGA LGG tumor samples via GDC and Xena. Normal brain reference transcriptomes from the GTEx project, accessed using a bioconductor package. We selected 150 GTEx samples spanning 12 anatomical brain regions, including cortex, frontal cortex, hippocampus, cerebellum, amygdala, among others, using a fixed random seed for reproducibility. It is important to note that the somatic mutation analysis was performed on the TCGA LGG tumor samples. So for the batch effect correction using combat-seq, so batch correction deserves its own slide because it is not optional here. So we use combat-seq rather than standard combat because RNA-seq counts follow a negative binomial distribution in this data set, not a normal one. Combat-seq models use counts directly using negative binomial regression, removes batch-associated variance, defined as TCGA versus GTEx data source, while preserving the tumor versus normal biological contrast needed for differential expression. One caveat that I would like to add, GTEx postmortem brain tissue is biologically different from intraoperative TCGA tumor tissue. So the combat-seq removes technical artifact, but it cannot remove that tissue origin biology entirely. This is acknowledged as a limitation and it should be considered when interpreting expression contrast. So the DSeq2 workflow. So the differential expression was run using DSeq2. The parameters included size factor normalization by using the median of ratios method, wall test for log2 fold change estimation, width shrinkage for low count genes, and Benjamini-Hochberg FDR correction across all 51,001 tests. Our thresholds adjusted p-value below 0.05 and absolute log2 fold change of at least 1. So the result, 86.3% of all tested transcripts reached statistical significance This figure warrants careful interpretation. While unusually high, it is consistent with the genome-wide epigenetic remodeling reported in IDH mutant glioma by Malta 2018 and Lu 2012, and reflects pervasive transcriptional divergence between tumor and normal brain rather than analytical inflation. Under stringent thresholds, 25,647 genes were qualified as significant differentially expressed genes. The directional split, which is 25,051 downregulated versus 596 upregulated, a 42:1 ratio. We move on to the results. So starting with the mutation overview, across 510 tumor samples, 96.447% harbored at least one somatic variant, which is an extremely high penetrance. The average was 28 mutations per sample. Missense mutations dominated at 65 to 70%, consistent with the IDH hotspot architecture. The transition to transversion ratio of approximately 2.3 at the bottom left corner, above the somatic baseline of 2.0, is already indicating that C to T transitions are dramatically enriched, that I will explain shortly. So the next 3 slides basically consolidates the 3 lollipop plots that I will describe shortly, together with the somatic interaction matrix. So for IDH1, is mutated in 70% of this cohort, which is 395 out of 510 samples. Of those, 394 carry substitutions at arginine 132. So the mutation is localized at the R132H region for 90% of the cases. Arginine 132 sits in the active site of pocket of IDH1, where normally coordinates the isocitrate substrate. The R132H substitution prevents isocitrate binding and redirects enzyme activity towards producing D2HG that was mentioned earlier. The fact that 394 out of 395 IDH1 mutant tumors hit the same codon is diagnostic of a neomorphic gain of function mechanism rather than random disruption. This is also why vorasidenib, which targets this exact pocket, represents a genuine therapeutic advance. Coming to TP53, TP53 mutations were found in 48.63% of the cohort, which is 248 out of 510 samples. They concentrated in the DNA binding domain between residues 100 and 300, accounting for approximately 75% of all TP53 mutations. 2 mechanistically distinct types operate at this position. Contact residues, which is at R248 and R273, directly block sequence-specific DNA binding. Structural residues R175H and G245S alter the conformational fold of the DNA binding domain, producing both loss of function and gain of function effects simultaneously. The strong co-occurrence of TP53 and ATRX mutations at p-value below 0.001, as I will demonstrate later, confirm the astrocytoma lineage tract of IDH mutant LGG and its mutant mutual exclusivity with the oligodendroglioma lineage markers CIC and FUBP1. For ATRX gene, it's qualitatively different from both IDH1 and TP53. There is no hotspot. Mutations, as you can see, are distributed across the full 2,492 amino acid nucleotide amino acid peptide, affecting the ADDZ chromatin reader domain, the DEX, DC, and DEAH helicase domains, and the helic domain. This is a distribution of a tumor suppressor gene being disrupted, not an oncogene being activated. Frame shift indels and nonsense mutations together account for 55% of ATRX mutations, truncating variants that introduce premature stop codons, triggering nonsense-mediated decay, and producing functional null alleles. ATRX is not mutated into something new, it is simply absent. The consequence of it is disruption of chromatin remodeling at repetitive genomic regions, enabling ALT telomere maintenance and amplifying epigenetic silencing of clustered gene families. This is the somatic interaction matrix of the TCGA samples. So the stronger co-occurrence signal is between IDH1, TP53, and ATRX, which are the three dark green cells in the upper left column. IDH1 and TP53 co-occur significantly, and TP53 and ATRX co-occur significantly at a p-value below 0.01. This triangular co-occurrence pattern defines the astrocytoma lineage tract of IDH mutant LGG and is consistent with the established molecular taxonomy of WHO classified IDH mutant astrocytoma. CIC, which is the fourth row, and FUPP1, which is the sixth row, show significant co-occurrence with each other and with IDH1, reflecting the oligodendroglioma lineage, where CIC and FUPP1 mutations accompany IDH mutation and 1p19q co-deletion. Notch 1 also shows co-occurrence with this group, consistent with its known enrichment in the oligodendroglioma subtype. The mutual exclusivity signal, shown in brown and orange, is visible between the astrocytoma lineage markers and the oligodendroglioma lineage markers. ATRX show mutual exclusivity with CIC and FUPP1, and TP53 shows mutual exclusivity with CIC, confirming that these two molecular tracks of LGG evolution are largely non-overlapping in the same tumor. This mutual exclusivity is the molecular basis of the two-track model, as mentioned earlier in the WHO classification, and is confirmed here at cohort scale using statistical testing rather than clinical annotation alone. Next, we will go to the PCA analysis. So the principal component analysis was performed on the log2 transform post-combat seq-corrected expression matrix of 684 samples from both the normal and also the tumor samples. Panel A shows the scree plot, whereby PC1 explains 10.3% of the variance and PC2 explains 5.5%, and the curve flattens rapidly after PC3. Approximately 276 principal components are needed to reach 80% cumulative variance in this cohort. Panel B shows the PC1 versus PC2 scatter plot. Tumor and normal samples occupy broadly distinct regions of component space. Panel C shows pairwise scatter plots across PC1 to PC7. I want to address the low PC1 variance directly because it might appear to suggest that the analysis is underpowered, but I would like to emphasize that it is not. A PC1 of 10.3% is the expected result when 4 distinct sources of variance compete simultaneously, which is the molecular subtype heterogeneity, variable tumor purity, residual multi-institutional technical variation, and genuine within subtype biological heterogeneity. These seek to test each gene independently and is not sensitive to the multidimensional PCA variant structure. So coming to the downregulated versus upregulated ratio, 25,051 downregulated genes versus 596 upregulated genes, a 42:1 ratio that reframes how we should think about lower grade glioma biology. LGG is not primarily a disease of oncogenic activation, it is a disease of transcriptional erasure. The IDH G-CIMP axis removes programs rather than adding them. This has a direct therapeutic implication. Strategies that aim to restore silent programs, IDH inhibitors, DNMT inhibitors like azacitidine, EZH2 inhibitors, are logically aligned with the disease mechanism in a way that simply targeting upregulated genes is not. The most biologically informative genes in LGG are the ones that are absent. So coming to the KEC pathway enrichment, GAGE analysis across the 177 human KEC pathways identified exactly one genome-wide significant pathway, which is the HSA04740, connoting the olfactory transaction with a p-value of 2.57 times 10 to the power of negative 45 and Q value of 4.21 times 10 to the power of -3, which indicates a genome-wide significance after the BH correction. All other 176 pathways that were tested were nominally significant at best, and none of which survived genome-wide false discovery rate correction. So a Q value, as mentioned, of 4.21 reflects the near complete coordinate suppression of 323 OR, which is olfactory receptor genes, simultaneously. That scale is only achievable through genome-wide epigenetic silencing of chromosomal domains under G-CIMP. The OR pathway Q value may therefore serve as a continuous quantitative indicator of G-CIMP severity across patients. So coming to the biomarker prioritization, to identify biologically meaningful candidates from the 25,647 significant genes, we use a two-step prioritization. Step 1, take the stringent DEG's gene symbol list and compare it against the CTG, the comparative toxicogenomics database, glioma gene set, genes that were independently curated as glioma relevant under the mesh descriptor D005910 using Veni. The intersection produced IFNA6 as the top candidate. So it is type 1 interferon gene cluster, and it met with 3 simultaneous criteria in our study, which is statistically significant in our DEG analysis, independently validated by the CTD disease annotation, and located at a genomic locus with established biological relevance to the cancer immune evasion. Right, so allow me to explain briefly regarding IFNA6. So basically, this is a schematic map of the chromosome 9p21.3 locus plotted along the HC38 genomic coordinate axis. This spans approximately 21.2 to 22 megabases. The x-axis represents physical chromosomal position in megabases, and the y-axis represents gene tract position along the chromosome. 4 gene types are color-coded in the legend at the bottom. So the CDK inhibited genes are shown in red. These are CDKN2A and CDKN2B, positioned at approximately 22 megabases on the right side of the figure, as you can see at the bottom right corner. IFN alpha genes are shown in blue and constitute the largest gene family on this locus, which is IFNA1, IFNA2, IFNA4, 5, 6, 7, 8, 10, 13, 14, 16, 17, and 21, which are all visibly clustered approximately 21.4 and 21.7 megabases. The IFN beta, shown in purple, sits furthest to the left at approximately 21.2 megabases. The IFN omega, as shown in teal, which is the IFN W1, is positioned between IFN B1 and the IFN alpha cluster at approximately 21.3 megabases. The orange dash box, as you can see, highlights the IFNA6 genomic position and is situated at the 21.55 megabases cluster. Gene directionality is shown by arrows on each gene tract. The majority of IFN alpha genes are transcribed in the same orientation, consistent with their shared evolutionary origin as a tandemly duplicated gene family. The key spatial relationship illustrated by this figure is the proximity of the type 1 interferon green gene cluster to the CDKN2A and CDKN2B tumor suppressor loci. Although IFN A6 and the broader IFN alpha cluster sit approximately 400 kilobases upstream of CDKN2A, focal deletions originating at the CDKN2AB core in malignant glioma can extend leftward along the chromosome to encompass the IFN gene cluster, providing the genomic basis for the co-deletion mechanism that links CDKN2AB loss to IFNA suppression in glioma, as demonstrated by James 1991 and Barriga 2023. So the IFNA indeed seeks to statistics. So it had a log2 fold change of -1.01 corresponding to approximately twofold lower expression in lower grade glioma tumor relative to normal brain. The adjusted p-value is about 3.94 times 10 to the power of 7, negative 7, which is indicating of genome-wide significance after BH correction across the 51,001 transcripts. IFNA6 is not the most strongly suppressed gene in this dataset. Many olfactory receptor genes that were affected in the pathway I mentioned earlier have larger fold changes, but its significance comes from the convergence of the three methods and evidence streams that I demonstrated earlier. So we move forward to the discussion. So the central intellectual contribution of the study is the 4-step mechanistic chain, which is step 1, the IDH1R132H demonstrated by the mutation analysis. Step 2 is the G-CIMP induction through TET2 and KDM inhibition, which indicates genome-wide epigenetic remodeling. Step 3 is the transcriptional suppression at a scale not previously quantified, which is a 42:1 ratio, and the 323 gene OR pathway silencing. Step 4 is the IFNE6 silencing at 9p21.3, which is a type 1 IFN pathway loss, which indicates immune evasion and checkpoint resistance. This chain, from a single point mutation to immunotherapy failure, has not been articulated with this degree of genome-wide quantitative support within lower grade gliomas. The clinical translation is the operational hypothesis using the 9p21.3 deletion status and IFNA6 expression to stratify patients before checkpoint immunotherapy trials. Why does IFNA6 suppression matter so specifically? Because type 1 interferons control multiple arms of anti-tumor immunity simultaneously. They upregulate MHC class 1 and 2 on tumor cells and antigen-presenting cells. They drive dendritic cell maturation. They activate the ISG program, which is IFIT1, MX1, OAS1, and STAT1, which is anti-proliferative and pro-apoptotic. They polarize macrophages towards the M1 anti-tumor phenotype. And critically, as LIM 2024 demonstrated in a single-cell RNA-seq of glioblastoma, that type 1 IFN signaling is the dominant communication axis between myeloid and T cells in the tumor microenvironment. Losing IFNA6 does not just reduce one signaling molecule, it collapses the entire immune surveillance architecture. So a few analytical limitations that I would like to address in the study. First, bulk RNA-seq captures a mixed cellular signal. IFNA6 is expressed primarily by the plasmacytodendritic cells and macrophages rather than the tumor cells. So its suppression in bulk data may partly reflect immune cell depletion rather than tumor cell silencing. Secondly, the comparative toxigenomic database-based prioritization selects annotated genes, not functionally validated ones. Third, we have not performed survival analysis linking IFNA6 expression to patient outcomes. Fourth, the mutation cohort of 510 samples and the expression cohort of 534 samples are overlapping but not identical. Direct mutation expression correlation was not performed within this analysis. And fifth, the combat-seq corrects technical batch effects but cannot eliminate tissue origin biology difference between postmortem GTEx and intraoperative TCGA samples, as mentioned earlier. So conclusion, the key findings, which include the 4 principal findings from the study. 1, the somatic mutation landscape, which is the IDH1, TP53 and the ATRX, precisely recapitulates the canonical 2-track LGG model and validates the analytical pipeline. 2, the 42:1 downregulation symmetry across 25,647 genes, a normal quantitative measure of G-CIMP transcriptional dominance not previously reported at this resolution. Third, olfactory transduction as the sole genome-wide significant CAC pathway at a significant Q and P value, a quantitative readout of G-CIMP severity. 4, the gene IFNE6, which is a high confidence biomarker candidate connecting IDH-driven epigenetics to immune evasion and immunotherapy resistance. So scholarly implications, 3 fields can benefit from these findings, which includes molecular oncology, whereby the IDH mutation establishes a genome-wide immune exclusion phenotype. So the 40:1 ratio reframes the biomarker discovery lens entirely. For clinical trial design, 9p21.3 deletion status and IFNE6 expression emerge as candidate predictive biomarkers for checkpoint immunotherapy certification, analogous in clinical logic to BRAF status in melanoma. For computational oncology, this study demonstrates that public multi-omics datasets, when integrated through principal batch correction, rigorous statistical testing, and disease knowledge cross-referencing, can generate mechanistically actionable hypotheses without any new experimental data collection. The present findings also establish 5 future directions following a logical sequence. First, resolve the mechanism of IFNE6 suppression using TCGA GTEx2 and 450K methylation array data already available through GDC. Second, demonstrate clinical relevance through the Kaplan-Meier survival analysis and Cox regression using the TCGA clinical metadata. Third, resolve cellular specificity using timer 2.0 deconvolution applied to the existing TCGA LGG expression matrix. Fourth, confirm the IDH G-CIMP IFNE6 causal link in experimental models. And fifth, independently replicate the IFNE6 downregulation across CGGA, Rembrandt, and GEO cohorts. Each step is tractable within existing data resources or standard experimental frameworks. So to summarize in 3 sentences, lower grade glioma is defined by what is absent, a 42:1 downregulation ratio that reflects CGMP erasing the normal brain transcriptome rather than oncogenes taking over. IFNE6 connects that epigenetic mechanism to immune evasion in a way that is mechanistically coherent, statistically significant, and therapeutically actionable. And the 9p21.3 deletion status is a testable stratification criterion for checkpoint immunotherapy trials, one that could distinguish patients who are likely to respond from those who need IFN pathway restoration first. Thank you very much.